This is what we did with Marianne Fyn and Sturla Molden, our two students, and then still in collaboration with Menno Witter. So this shows a rat brain from behind. This is the brainstem. Entorhinal cortex is the colored region here, and this is the medial part. And the electrodes were put up here in the really in the dorsal part, which is the area where cells project directly into the place cells in the hippocampus. So recording in this area, this is what we observed. So 2 seconds. Now you see a rat running around, chasing chocolate in this box here. And whenever this cell that we are recording from is firing an action potential, then we see a white dot here. So the cell is active here, there, but it's active at many places. So in that sense, it's quite different from the place cells that John showed. Now we just speed up a little bit. And at first glance, somewhat disappointing, because you can see firing everywhere. But as you look a little bit more, you'll see that actually there are many firing fields, but they each are quite discrete. So they sort of stand out from a background of no firing. And the other thing that you may notice is that not only do we have multiple fields, but they actually are quite regularly spaced. So this intrigued us. We didn't quite understand that regularity at that time yet, but soon after it became clear. And what we did here is that we let rats walk in a larger environment. In this case, I show it for a very large one is 220 by 220 centimeters. And the gray trace here is the path of the animal in the background, and each black dot is the firing location of the cell. And you can really see how regular this is, that the pattern forms a grid that tessellates the environment, and for that reason we then call them grid cells. So this is work that we